This figure is an absolutely brief figure that just summarizes lipid-soluble hormones. The biochemistry of the lipid-soluble hormones are discussed in Chapter 5 in molecular biology because lipid-soluble hormones work at the gene level, and in Chapter 5, that's where regulation of gene expression is discussed, gene expression caused by lipid-soluble hormones. But briefly, in this figure, lipid-soluble hormones go through the plasma membrane, they come into the cell and bind to a receptor class called the zinc finger receptor. When the hormone binds to the receptor undergoes a conformational change, opens up in fingers, and then the protein, the receptor, goes in the nucleus and looks for a piece of DNA called the enhancer, and a part of the enhancer is called a response element. So the zinc finger protein binds to the response element of an enhancer region, and then the DNA bends, RNA polymerase comes in and turns on a specific gene. Now in Chapter 5 in molecular biology, that was discussed with a specific example in gluconeogenesis, where the gene that's being turned on is PEP carboxykinase in gluconeogenesis. And what's turning it on is cortisol, a lipid-soluble hormone, comes into the cell, looks for the zinc finger receptor for cortisol, which goes in the nucleus of the hepatocytes, looking for an enhancer region, and the response element is called the cyclic AMP response element. The DNA bends, RNA polymerase comes in, and turning on a specific gene for PEP carboxykinase. So then the gluconeogenesis is turned on. At the end of Chapter 9, there are 3 questions that I want to go over, questions 2, 3, and 4. In my opinion, what I would suggest doing is stopping the video, do these 3 questions on your own, and then come back to the video to obtain the answers. It would be to your benefit to go through that way.
This is a 2-part question. In the first part, it says that tumor cells from a person with leukemia have been analyzed to determine which oncogene is involved in transformation. And it says, after partial sequencing of the gene, the predicted gene product is identified as a tyrosine kinase. And it says, which of the following proteins would most likely be encoded by an oncogene and exhibit tyrosine kinase activity? And the answer is option E, growth factor receptor. Remember, a tyrosine kinase is a receptor for a particular hormone. And in this case here, it says a tyrosine kinase, so you look for the word receptor. And of the 5 options, the only option that has the word receptor is growth factor. So that's why the answer is E. Now option D, platelet-derived growth factor, or option B, epidermal growth factor. They are hormones that bind to a receptor, but they are hormones, and the hormones do not have tyrosine kinase activity. In the next part, it says a kinetic analysis of tyrosine kinase activities in normal and transformed cells is shown below. So you see the Lineweaver-Burke plots of 2 enzyme activities, one's from normal cells and one's from tumor cells. Tyrosine kinase activities in the Lineweaver-Burke plot. And it says, which of the following conclusions is best supported by the results? And the answer is option D. A kinase gene has been amplified in the tumor cell genome. Notice on the Lineweaver-Burke plot that the Km value of the enzymes in normal and tumor cells is not changing in the tumor cells. But the velocity, the Vmax on the y-axis, is changing. And in normal cells, you see the value that's intersecting with the y-axis, and right below it is the value from the tumor cells. But the key here is going from top to bottom. If you increase the number of proteins, you're increasing Vmax. So the value in normal cells, that's a smaller value than in tumor cells. One more time. The Vmax in normal cells is less than the Vmax in tumor cells. And the reason is it's a double reciprocal plot. So going from top to bottom, you're increasing Vmax. Going from top to bottom on the y-axis, normal cells to tumor cells, You're increasing Vmax. And so, if you're increasing Vmax, that means the gene is now being amplified to make more enzymes. More enzymes. So tyrosine kinase activity is now increased, and that's due to more enzymes, and more enzymes can be derived by having more genes. So you increase the genes in tumor cells, increase the tyrosine kinase enzyme, and thereby increasing velocity.
Question number 4. In a DNA sequence project, an open reading frame has been identified. The nucleotide sequence includes a coding region for an SH2 domain in the protein product. This potential protein is most likely, answer to this question is option C. Transmits signals from a tyrosine kinase receptor. The idea is SH2 proteins are involved with tyrosine kinase pathways here. So SH2, you think tyrosine kinase. Now, once the SH2 proteins dock with IRS1, which in turn docks with the tyrosine kinase receptor, signals are being transmitted all the way to turning on gene expression. Now, why is option A incorrect? Because option A deals with specific transcription factors binding to an enhancer region in the DNA. So it has nothing to do with SH2 proteins. SH2 proteins are upstream. Option B, a transmembrane hormone receptor. Why is that incorrect? Because that's the adrenergic receptor, the serpentine receptors for epinephrine and glucagon, which have nothing to do with tyrosine kinase receptors. Option D, bind to an upstream promoter element. That's the general transcription factors. Remember in Chapter 5 in molecular biology, general transcription factors bind to TATA boxes, UPE, GC-rich boxes within the promoter. And in the promoter, general transcription factors, not specific transcription factors, bind. Lastly, option E, activate a soluble guanine cyclase enzyme in the vascular smooth muscle. Why is that incorrect? The reason is because that's what nitric oxide does. Nitric oxide comes in, as being generated, comes into the cell, activates guanine cyclase to make cyclic AMP to turn on protein kinase G to cause the vasodilation. Nothing to do with SH2 domain proteins.